छः।
A very good afternoon to all the participants present over here. Today is the fourth day of our this webinar, which is in the series of Faculty Development Program organized by Department of Entomology and Department of Plant Pathology, MS Swaminathan School of Agriculture, Centurion University of Technology and Management, Odisha. Today, our speaker is Dr. Chandrasekhar Prabhakar sir. He is an assistant professor in the Department of Entomology, Bihar Agricultural University, Sabor. Sir, I welcome you on the behalf of our university. In very short notice, you agreed to deliver lecture for this webinar. We are very much thankful to you, sir. Now, sir, please you share your screen and start your talk, sir. Before the talk of sir, I would like to announce that feedback form and for certificate download. The link will be available in today's session between any time. So participants are requested to please fill up the feedback form and download their respective certificates. The link will be shared in Zoom link, YouTube link, and all the WhatsApp groups. Now I request Prabhakar sir to please start his talk. Chandrasekhar Prabhakar sir. Hello. Hello, sir.
Hello, Ritesh. Are you getting my voice?
Yes, sir. I'm getting your voice. Please share your screen, sir.
Okay. Is some issue with the video? I am just checking here, but that is video is closed from your side. Zoom meeting is giving some problem here in my desktop. It's not working properly.
Hello.
Hello.
Prabhakar sir, is there any problem?
Yeah, it's a video is not getting on from here,
Sir, you can share me. I will share from here.
and share option is also. I'm sending you the file there.
Okay, sir. Okay. Hello. Hello. Hello, sir.
Visible.
Hello. Prabhakar sir. There is some monsoon and weather problem there. That's why sir is network issue. We are resolving in just 2 minutes. Uh, Hello, Prabhakar sir.
Hello.
Yes, you can continue, sir, please.
Yes. Are you getting my voice? Yeah. Thank
I'm
you,
sharing your
Ritesh.
screen from here, sir.
Yeah. And good afternoon to all of you, faculty members and students. So today talk is on diversity, spread and management of fruit flies in India. Actually, fruit flies are very important pest species or pest, a group of insect pests that actually control all the export of vegetable and fruits, mainly mango and other fruits and vegetables, mainly cucurbits. So next slide. So with the overview of fruit flies, uh, that is belongs to the order phylum Arthropoda, class Insecta, order Diptera, and the family of the fruit flies is Taphrytidae. Generally, people confused with the Drosophila. That fruit flies is Drosophila. So these are all fruit flies who actually the pest of different commercial crops are not from the family Drosophilidae. These are all from the family Drosophila. Taphrytidae, and these having a almost 481 genus and more than 5,000 species across the world. So in there is a 5 photographs in this slide. that is Ceratitis capitata, Regulatis pamelina, Decus asphoralis, Anastropha ludens, and Jugodecus cucurbiti. So in these are all are the very economically important fruit fly species. Among these species, only Jugodecus and Decus species and Bactocera species are present in India, and Ceratitis capitata, Regulatis, and Anastropha are not present in India, but they are very having very much threat to the Indian economy if entered once in India. So these are the quarantine pest for India. Next slide. So how fruit flies is important. Sometimes we find in newspaper that Indian mango or Indian vegetables are banned from the export from different countries. The recent news in 2020, the New Zealand bans Indian import of Indian mango for the when they find the fruit flies in the consignment. So in 2014, the European Union's banned fruit fly fruits, mainly mango and cucurbits, bitter gourd, banned for the export from India because they find fruit flies lay eggs and larvae inside the fruits. So these fruit flies are basically that control the international business of fruits, fresh vegetables, and fruits from different countries. So next, why fruit flies is important is international trade of fruits and vegetables, because when we look into the life cycle of fruit flies, this is Bactrosa tau that is generalized for all fruit flies. So the gravid female lay eggs inside the fruits, and then the small tiny white colored eggs that is laid inside the fruits, and larvae emerge from that eggs, remains inside and eat inside the fruits. And after maturity, the larvae come out from the fruits and then pupate inside the soil. And then from pupa, the small tiny insects, that is male or female fruit fly, emerge from the pupa and then forages for their resources, and again mating, and the cycle continues in that way. So what happened when exporting the fruits? When exporting the fruits, at that time we don't know that fruit fly eggs are inside the fruits, or larvae inside the fruits are there or not. At the time when we send the consignment, and the difference, the time difference between the consignment sending and the consignment receiving at that point in other country, there's just 4 to 5 days gap. In that time, the from eggs, the larvae hatched out, and a smaller larvae become larger or bigger. So in that way, the that receiving country find there is a infestation of fruit fly, but sending country didn't find that is infested from that. So this gap of 4 to 5 days, that basically create the issue, that development of fruit fly larvae and eggs. And that time, and if the country is not having that particular pest, a species of fruit fly, then they cancel entire consignment and they return, or they destroy the entire consignment at their port. So they don't want to allow other fruit flies. So next, so this is the actually one more tab. Enter. Yeah, this is the geographical distribution of fruit flies. So in that case, there is a 5 different geographical areas, 7, that is Nearctic, Oceania, Neotropical, then Palearctic, Afrotropical, and then Oriental, Indo-Malayan, and Australasian area. So on that geographical basis, in India, we find around 100,000 species of fruit fly is in Southeast Asia and India together, and there is a 155 genus. So they have fruit flies distributed among in entire world in different species and species diversity. So in India, basically Bactrocera and Juvederm are the main fruit fly species. So I'm coming to the history of fruit flies in the India research. Next slide. Yeah, history of fruit flies in India. So the with the establishment of Imperial Institute of Research, Agriculture Research Institute at Pusa, Bihar, most of the work started in the that time of Bihar and the entire India. Basic work of entomology started with the present Bihar, where that institution are established at that time. And in that, Beji reported different fruit flies work during 1913 and 1916. And after that, a big gap in fruit fly research, and Kapoor and Agarwal did vast work in the fruit fly fauna of North India. And then Raghu and Drew worked in the South Indian fauna of fruit fly. And the methyl is a very good attractant, parapheromone, that is discovered, the methyl property of fruit fly attractant, discovered by Frank Halbert in 1912 in that time of Bihar. And till today, the methyl is a very good source of attractant of fruit flies. That is male fruit fly, basically generally Bactrocera dorsalis, Bactrocera jonata, and Bactrocera correcta. Next. So in the Indian fruit fly diversity, there is a 3 subfamily we have reported from India. We already have in India, and 18 tribes, and that distributed in 79 genera and more than 250 species. But we think about that 8 to 12% of world diversity of dipterans are present in India. If we calculate or estimate on that basis, more than 400 fruit fly species are estimated from India, and still discovery are going on the fruit fly, different fruit fly, new fruit fly species from India. So I'm coming to the major economically important fruit flies in fruit crops in India. That is next. Next. So this is the Bactrocera dorsalis. This is one of the most important fruit fly species across the world, and this is actually called as the Asian fruit fly or mango fruit fly, sometimes Oriental fruit fly. So different common name, but one scientific name, that is Bactrocera dorsalis, and first described by Hendel from Taiwan in 1912. And in India, first described by Beji in 1913 from Bihar. And origin of that species at that time is not known, but now it's originated in South Asian countries, maybe India, Bangladesh, Nepal, but most probably from India. And this is very polyphagous, very much polyphagous insects, and having reported 450 host plants. So think about that, how much damage it may cause. The most fruit fly, most fruit species or crops that is infested by these particular species, and widely distributed in Asia and Africa. This species is not present in Europe and American continent and Australia. But recently, this reported from the Italy, one port of Italy. So there is a very huge surveillance is going on for this species in the European continent for this species. So there is a many cryptic species. Cryptic species are those species that is not easily identified with the morphology. They look similar but having different mating and behavioral and host classic host species. So on that basis, the major host of this species in India are mango, guava, sapota, lychee, and peach, plum. These are the major host plants. So next, yeah, coming to the invasion, go back. In 2012, there is a study from Chinese authors, researchers. They came that the fruit fly came in India from Southeast Asia, and that time they used Himachal Pradesh data on cytochrome oxidase sequence, and that basis they concluded that from in India the fruit flies Bactroceras dorsalis came from Southeast Asian country. When next, when go back to the distribution of Bactroceras dorsalis complex, I already told you that there is a 4 species in Bactroceras, many species, but 4 very common species and 4 different names. That is Bactroceras invadens, that is present in the that is African continent, and the Bactroceras philippanensis, that is present in the Philippines, and Bactroceras papai, that is present in the Southeast Asian country, and Bactroceras dorsalis, that is present in mainland of Asia, that is China, India, Bangladesh, and Pakistan. So on that basis, these are the 4 species, and every country having quarantine restrictions on the import of fruits from those countries. So they look similar, but they have different restrictions. So the world community of fruit flies who is working on fruit flies, they work together for more than 20 years and published a paper during 2014 that all the 4 species, that Bactroceras dorsalis, Bactroceras philippanensis, Bactroceras papai, and Bactroceras dorsalis are the same species. They are not different. So they synonymize the name of the species and keep the valid name as Bactroceras dorsalis. What happened after this? Next. So in the 20 years, they studied and they find that all the species are 4 species are same, and they kept the name Bactroceras dorsalis for. And after the synonymization, the fruit export and import of fruits from different countries are very easy because all different countries having the same species. So we can easily send the consignment to other countries who having this particular pest in their country. So next. So what happened after synonymization? Synonymization having given 2 questions: where the original origin of that particular Bactroceras dorsalis, and how they are spread from different area. Are they spread from Africa to India or India to Africa, and vice versa, like that. So we studied one study we did in 2000 and published in 2016, the genetic analysis of oriental fruit fly Bactroceras dorsalis. We collected samples from different states of India, and we also collected the sequences of CO1 and NAD1 gene, mitochondrial gene, from the NCBI database. Next, we collected the samples from Indian different state of India, and we also collected the sequences of COX1 and NAD1 gene from different countries, Asian countries, because that time they're present in Asia only. So after that, we find that Next, there is a this is a haplotype network that is based on that single mutational step. If you find there is a link between the two haplotypes, so that is maybe single or double mutations. So we find that there is a huge haplotype diversity, but they are not forming any differentiation in the population. Population differentiation means are the different populations sometimes are really having much genetical differences from the other population. So we didn't find any much uh, mutational differences in the genetic sequences, gene sequences of NAD and CO1 of Bactrocera dorsalis population from Indian and Asian countries. So we find that there is no significant population differentiation of the Bactrocera dorsalis species. So this conclude that we can manage the population with the help of different genetical tools. I'm not going to tell that discussion is the different genetical tools in this presentation, but the species is same and all the population having similar genetic similarity. So we concluded in this study. After that, we also I was the part of one international collaborative research project on population structure of a global agriculture invasive pest, that is Bactrocera dorsalis, and we find here that the origin of that particular Bactrocera dorsalis is South Asia, and from South Asia there is a two route. One is in one movement of this fly is from Southeast Asia to the Australia and China, and second route is from Africa to the Hawaii. So these we can find we find in this particular research we did in collaboration with the Chinese authors, and among 20 countries samples we used for this study and published during 2018. Now coming to the Bactrocera jonata. Next, this is the peach fruit fly, Bactrocera jonata, and in jonata that is also first time described in 1842 by the Saunders in then Bengal, from with the name of Desinura jonatus, and this also having probable Indian origin, and we need that kind of a study what we did with that Bactrocera dorsalis, and this is also distributed in Indian and Southeast Asia, and recently invaded to the African country. So there is a this is also a polyphagous pest, and established in Egypt in 1990. So we did a mitochondrial genome sequencing. Next, we did mitochondrial genome sequencing for that. If any there is any multi multi-gene phylogenetic study, and we published this paper in Gene 2000 during 2015, and we found that there is a no cryptic species found in this particular Bactrocera jonata. So Bactrocera jonata doesn't have any cryptic species. All Bactrocera jonata having similar genetic and morphological appearance. Then next, so coming to the guava fruit fly, that is Bactrocera correcta, and this is also reported first time from the Beji in 1906 from Bihar, and that is well distributed in widely distributed in South Asia and Southeast Asia, and they have reported 62 host plants from 30 plant families. So this is also a polyphagous pest, and couch In the economic importance, this is on the third rank after Bactocera dorsalis, Jonata, and the Correcta. And the next, and we found one study from the Chinese author that Bactocera correcta having some genetic differentiation in different population, and they found that there is two lineage. They have some where some specimens they found they have very wide genetic dissimilarity with the main population. So we need to check out in the India also that what lineages are present in India right now, or is there is a mixture of lineages in India or genetic differentiated population present in India. So these questions we need to address in the coming years, that there is no study from the India as I know till today, the no study of Bactocera correcta population genetics from India. So we need to work on it. Next. So coming to the spread of fruit flies infesting fruit crops in India, there is a three species. I already told about the population structure and different international status of that species. Coming to the India, what is the status of these fruit flies who is infesting fruit crops in India? So the circle is for Bactocera dorsalis and triangle is for Bactocera jonata, and the square is Bactocera correcta. When it's coming to the population level, so red color designated the high population intensity, and green color designated the low population intensity, and the black color designated the we don't know exactly the what is the population status of these species in the these species of fruit fly. So we find that high population of Bactocera dorsalis found in the southern India. And when we talk about that high population of Bactocera jonata we found in the northern India for fruit infesting. But low population we found everywhere of all these three species, that is Bactocera jonata, Bactocera dorsalis, and Bactocera correcta. But a gap is we don't have any data from the central India, that is MP, Chhattisgarh, and nearby the Rajasthan, and the Northeast Asia on the population dynamics and population intensity. So nowadays the Chhattisgarh and MP is coming up with the huge horticultural orchards, and maybe there is a problem of fruit flies, but may we get the data in the coming years from there, the population dynamics and population intensity. So on that basis we can see here that Bactocera jonata is more severe in the northern India, and Bactocera dorsalis is the more severe in the southern India, and Bactocera correcta is the present everywhere but in low population. Next, I'm coming to the vegetable infesting fruit flies. Next. That is melon flies, that is Zygotacus cucurbiti. The recently name changed from Bactocera cucurbiti to Zygotacus cucurbiti. There is a earlier this species name is Decus cucurbiti, then Bactocera cucurbiti, then Zygotacus cucurbiti. So now presently this species name, scientific name is Zygotacus cucurbiti. So this first reported from India, Bihar in 1913, and that I worked on this species during my PhD, and I find that this species is originated from India, and about 4 lakh years ago, and then widely distributed in Asia and Africa nowadays, and they are highly invasive and adaptive in a new climate, and these are a species having 125 reported host plants and major pest of cucurbits crops. Next. So we found it during 2008 that very less genetic diversity found in this particular species across the Chinese population, that studied by Hu et al. During 2008, and then Wu et al. Published 2000 during 2012 that some little bit more genetic variation they found in that with the mitochondrial gene analysis. And the same time I found that from the different countries on the analysis with the international sequences from other countries, we found only 2 mutational steps. We found 3 mutational steps. The entire population is with that 3 mutational steps. So there is not much variation in the that is differentiation of population, but yes, variation is there on the haplotype basis, not differentiation. So the genetic makeup of this particular species in India is entirely same, and we can control, or we can manage this species with the sterile insect techniques, or with the genetic manipulation. That is, we are right now we are going with that in different country with the genetic engineering tools. So the similar genetic makeup help to manage the population with the single genetic manipulation techniques. So this study only conclude that this is a highly similar population we found in India. Next. Coming to the next species, that is Jugodecus tau, and Jugodecus tau is also earlier name was Bactrocera tau. This is reported first from the 1849 from Fujian, China, and the most probably the Southeast Asian origin, and widely distributed in Asia. And there is the 8 cryptic species in Bactrocera or Jugodecus tau complex. More than 60 host plant has already been reported, and major pest of cucurbits. Next. So we studied that with the different collection from the India, we find that our species of Jugodecus tau complex, that is type A species. Jugodecus tau A is the species we find in India, and other species that is based on the sequence, that is Bactrocera tau B, C, D, E, F, I. There is 8 species already reported in the complex, but we have only one species, that is Jugodecus tau type A species in India, and this is the we studied during 2013. Next. We have another species that is cucurbits infesting, Jugodecus scutularis, that is Asian origin. We didn't find that from which country they are, this particular species are originated from. So, and very much damaging to the cucurbits. And if we found that this particular species are very damaging in the apical part of the crop cucurbits, or very small fruits. When fruits are tender, they infest, and they if the infestation of this particular species appears in the field, no, you can't find any mature fruits. All fruits will fall down or damage with this particular species. So this is very damaging a species, and you can't, and the control of this species is very important for the production of cucurbits in area where this particular species is present. This species we found in the traps in northern India, even Bihar, Himachal Pradesh, and Uttar Pradesh. So we have from 3 locations we have data of this presence of this species, and other areas also, maybe other region of other state of India may have this particular species. So this is also a important species, and we need to work on it on the population structure. So next. So coming to again the population intensity and population presence spread of these particular species who is infesting cucurbits in India. So Jugodacus cucurbiti is in a circle, and Jugodacus tau is the triangle, and Jugodacus scotralis represented with the square. And we have high population with the red signal color and low population with the green color. So we find that Jugodacus cucurbiti is present all across the India with the high population. Wherever you find the cucurbit crops, you may find this particular infestation of this Jugodacus cucurbiti species, and there is huge damage caused by these insects. And this is very Jugodacus cucurbiti is the very important for the export. We can't export bitter gourd because the finding the bitter gourd infestation of fruit fly in bitter gourd is very difficult. So on that basis, and we found that Jugodacus tau also present in across the India, but it's very low population. And Jugodacus scotralis we found high population in Himachal Pradesh and low population in Bihar and other area. So on that basis, the for cucurbits infesting Jugodacus cucurbiti is very important for entire area. For the hilly areas, Bacteria or Jugodacus tau is the very important species. Next. I'm discuss some other fruit fly species. These are the Bacteria latifrons, commonly called as Malaysian fruit fly or solanaceous fruit fly. These particular species, Bacteria latifrons, cause damage to the solanaceous crops like chili, then brinjal. So these two in the Southeast Asia or in Thailand, this particular species very much damaging to the chili, and this is and tomato also. So this particular species we found in India, and the major damage caused this particular species is in South Asia, South India, sorry, and in the we have the population of this particular species in Himachal Pradesh and Bihar also, northern India, but in low population. Next. And then Bacteria nigrofemoralis. This is we find this particular species very high population in traps in the northern India, but there is no significant. This is not a significant pest of crops, that is cultivable crops. So commercial crops of fruits and vegetables. Their known host, only one known host, that is Traminaria catappa, but the huge population we found in the traps, and that's why the trapping intensity is the not a we need to identify the proper species and then count the population, because if we found it that in trap the huge number of fruit flies coming and the high population of Bacteria dorsalis, that is the wrong statement. So we need to check which species is high and what are the host plant in of that particular species in the literature or in the area. So we need to report on that basis. Next. This is Bacteria diversa. This is also a cucurbit infesting fruit fly, and this feeds on flower of cucurbits and not a major species. We can, if we rear or we install the trap, we can find that fruit flies one or two in the season. So there is not a huge population, but yes, this feeds on cucurbits. This species feeds on cucurbits. Next, then coming to the Jugodicus cordata. Yes, these species are having different lineage described by in 2012. There is a two lineage. One is Indonesia and one is Malaysia. And in India also we found in the two there is a four reported locations. That is from South Asia, South India, and Western India from Mumbai, and from Eastern India, Bihar and Jharkhand. And we also found it in that Himachal Pradesh. So we studied that lineage of this species and published the paper in 2019. We found that two distinct lineages are found in the Indian, three distinct lineages found in the analysis. That two are the present in the that is already previously reported, but we found that there is a one that is entirely, the Indian population is entirely different. One, the Malaysian population is at other end, and the Indonesian population is the other end, and Indian population are the intermediary of these two populations. So we published this research in Current Science in 2019. Yes, next. Next. Now coming to the pest species. So these are the fruit fly species. I already discussed most of these species. When you take a look on these are very similar, look like very similar. Most of them you can differentiate when you look carefully, but they are look like similar. So for non-taxonomist who is actually working on entomology or pest management specialist, we did a barcode or CO1 genetic analysis and phylogenetic analysis. On that basis, can we separate these species on the basis of barcode, DNA barcode? So next. Yes, we find that this particular species population from different part of India, we collected the Bactrosa cucurbitae, Bactrosa tau, Bactrosa ascoturalis, Bactrosa jonata, dorsalis, nigrofemoralis, and Decus longicornis and Decus asperalis. And we found that they are the mitochondrial cytochrome oxidase gene 1 are very much helpful to separate these species with the help of genetic sequences of the particular gene, and they placed in the phylogenetic tree. They are separately placed. So if a non-taxonomist who is don't have much idea about the species, they can go for that molecular biology tools to use or correctly identify these species. They are very good source, useful tools for the species identification. Next, I'm coming to the management of fruit flies. Next. So why fruit flies management is very tough or complex. Fruit flies adults are very tiny and having the size of house fly and very active flyer, and all life stages are found in the protected place. I told you that in the life cycle, I already showed you that eggs are inside the fruits, then larvae are inside the fruits, and then pupa are inside the soil, and adults are very high flyer or very active flyer. So when you use the insecticide, you can't find that, you can't reach directly the insecticides to the eggs, larvae, and pupae. And whenever you use to find the adults, you spray the insecticides and adults will fly away. So this is the difficulty, and you can't use very toxic insecticides, very much that is pure poisonous to the fruit flies, because what we spray on that for the management of fruit fly, and most of the fruits we use for the table purpose. So we can't cook the fruits, we eat raw, and on that basis we can't use the very high or very poisonous, highly toxic insecticides for the management of fruit fly. So these are the limitations for the fruit fly control with the insecticides, and availability of all the stages at the same time. You can find the egg, larvae, pupae, and adults in the same field at during the season, and they are very much polyphagous in the nature. If crop is not there, they breed on the wild crops, wild fruits, and many generation in a year. That 9 to 10 generation, this fruit flies are having 9 to 10 generation in Indian condition. And if northern India is very cold, they move to the southern India and breed there, because the congenial conditions round the year are available in the southern India. Then short life cycle, they have only 15 to 20 days life cycle, and they move on, and complete more than 10 generation in a year. And host availability in kitchen garden, yes, we manage very carefully in the commercial cultivation, but we throw here and there in the kitchen garden, kitchen waste in the anywhere in the garbage. And also we find this host in the kitchen garden. So host available in the kitchen garden we didn't manage, we do not manage. So that increases the population. And one the high fecundity, because of the high fecundity means they one single female lay up to 300 to 500 eggs. So that one single female if alive, then population in the 2 or 3 generation, they become the high populated in that area. And they have very much resistant to the environment. They can stay in the hot summer and cold winter also, and favorable condition present in the entire India. So these are the complexities for the management of fruit flies. Next. So what are the current management strategies? So current management strategies are the area-wide IPM of fruit flies. Area-wide IPM means you can't manage at only one field. You must just try to manage in the area, entire area, like where you are, you have a 10 to 10 kilometer area of fruits, intensively cultivation of fruits or vegetables. You manage 10 to 10 kilometer. And for that management you use different components. There is just 6 components presently people using around the world for the management of fruit fly. They start from the population monitoring. We need to monitor the population around the year, the how, at what time, at what season they find the high population and low population. And the field sanitation, field sanitation is the very important for the fruit flies. And then weight spray are the spray with having some attractant and the insecticide. They spray on the fruits to manage the fruit flies. And male annihilation, they exclude the male from the environment with the to the objective of male annihilation is to kill the male with the pheromone traps. And Western countries and the Southeast Asian countries and Australia very much use in the Australian insect techniques. They are very much using these techniques for the control of fruit flies. They control actually the population of to remain low population, to keep fruit fly population low in the field. And biological control. Biological control presently used in the USA And in India, we only use 4 components: the population monitoring, field sanitation, a spray, weight spray, and male inhalation. We didn't use, or we do not use this particular astral insect techniques in India, and biological control for the fruit fly. Next. So in the field sanitization, literature say that if you keep the field sanitization properly, you can reduce the fruit fly population by 20% in the field itself. So these are the picture of very poor field sanitation. Fruits are fall down on the ground, and they kept there, and larvae emerging from there, and they go inside the soil, and adults come out. So on that basis, we can't manage that population of fruit fly. We need to keep field very clean. Uh, uh, Not a single fruit that is on the ground. So we need to infested fruit to the uh, kept in the pit, and spray the insecticide there to control. So field sanitization is very important component for the management of fruit flies. Then next. Then coming to the fruit bagging. Yes, fruit bagging of the host fruit is very good management options, but people are telling every time telling that this is very labor intensive, time consuming. But most of the export quality of fruits are come from the fruits bagging component, and in the Southeast Asia they are very much huge export of fruits from there. They all do the fruit bagging regularly in the field. So fruit bagging is the very good option for the fruit fly management. Next. Then coming to the weight spray. Weight is the an attractant and food source mixed with the insecticide to treating the fruit fly infested area. So there is two types of spray for the fruit fly management. One is the spot spray, and one is the cover spray. In most of the Indian literature, they use cover spray. Cover spray means you spray entire field for even for going for the fruit fly management. But in if you use the weight, you should not spray for the cover spray. Go for the cover spray. You use the spot spray. A spot spray means if you spraying in the orchard, you spray the alternate tree for the fruit fly management. So what happened with the alternate tree spray, that bit having some attractant in that. So from the neighbor's tree, the fruit fly attracted to the sprayed tree and get killed. So on that basis, in the cucurbits also, you can spray the one plot, a small plot. You just plotting in the field, and one plot is with the spray, and left one alternate plot without a spray. So on that basis, you can reduce what one is the labor, second the insecticide dose, and uh, so on that basis, you are saving the environment also and getting more profit. So weight spray is with the a spot spray only. And if you want to go for the cover spray, no need to wait. Mix the food source or food attractant. You spray simply the insecticide. Next. So coming to the development of a genetic male annihilation techniques for fruit flies. Actually, the fruit flies, I told you that fruit infesting a species, that is Bactocera caracta, Bactocera dorsalis, Bactocera jonata, and the fruit fly that infest cucurbits, that is Zygoticus cucurbiti, Zygoticus tau, Zygoticus caudata, Zygoticus Bactocera diversa. So there is two lure available in the market. One is the methyl trap, and second one is the Q lure. So who want to use to manage the fruit flies in cucurbits field, they must use Q-leaf. And who want to manage the fruit flies in the fruit crops, they must use methylaginol. But what happened in the field when farmers go to the insecticide shop, they ask for the fruit fly traps, and without asking the shopkeeper give the fruit fly trap, and they don't know which crop they are going to install. So, and sometimes farmers install fruit infesting methylaginol trap in the cucurbits field and Q-lure in the mango field. So there is a huge confusion in the market, and then not going to efficiently manage the fruit fly. So what we did, we experimented on the mixture of this both the lure that is available in the market, and we find that one good concentration that is attracting both fruit flies in a good attractant for the both the fruit flies. So that mixture we applied in the field, and we found that the good collection of the fruit flies and management of the fruit fly population. Next. So we have started uh, manufacturing that particular traps and septa for that particular fruit fly trap in Himachal Pradesh, and we tie up with the government of Himachal Pradesh, and we distributed in a very large scale. About 1 lakh traps has sold in the last 10 years, and so this is a very good model for the fruit fly management in the large scale area or larger area in different. And what happened with this, you can use that particular trap in the fruit crops or as well as in the cucurbits crop. So this is the one major things we need to do for the fruit fly management, that is mass trapping with the wide area management. Wherever you find the cucurbits field or mango or fruit crops, you need to install the traps. Next. So coming to one more recent area of research, that is Wolbachia indosymbiont induced cytoplasmic incompatibility for the fruit fly management. So this is called as IIT, incompatible insect techniques. This management technique is the incompatible insect techniques, and that is the Wolbachia is the bacteria that cause cytoplasmic incompatibility in the fruit flies. Next. So Wolbachia is having a very different manipulations in the reproductive system of insects, and they cause feminization in the different insects. They can cause male killing, they can cause parthenogenesis, but they can also cause these Wolbachia bacteria. They can also cause the cytoplasmic incompatibility. So the researcher that detected that cytoplasmic incompatibility in the insects, and that is also in fruit fly. So they try to use that particular behavior changing ability of Wolbachia, that is when Wolbachia infected male, female, and Wolbachia non-infected give the they will produce Wolbachia positive offspring. But in the vice versa, if the female is Wolbachia negative and male is Wolbachia positive, there is no offspring. They lay egg, but that is not viable. So this is the incompatibility, and this particular characteristics of Wolbachia people use in the pest control. What they do, they use to release Wolbachia positive male in the field, and whatever the Wolbachia negative female is there, if they mate, there is no population. So this is the very good and green solution for the pest management. So next. So what we did, we tried to find the Wolbachia population in different population of fruit flies and different insects of different species of fruit fly. So we found that there is a population that Bactocera cucurbitae or Jugozygus cucurbitae having much infection of Wolbachia from different part of India, and Bactocera dorsalis we find Wolbachia infection in the 3 species: Bactocera dorsalis, Bactocera cucurbitae, and Bactocera tau. And then we need to develop Wolbachia incompatible techniques in the future. So now next. So coming to the conclusion, and that is the fruit fly management. That is, fruit fly don't respect the farm boundaries, so need to a wide area management of fruit flies. Next. Next. So thank you. These are the different work I did in different area during my master and PhD and other sites. Thanks. Thank you so much. Any question?
Thank you so much, sir, for your such a nice and informative talk. Now I request the participants, if they have any queries, they can directly ask to sir. Any participants, if they have any queries, they can ask to sir. Or otherwise, I would like to request Deepan sir to please read the queries from chat box, sir.
Yes, sir. Thank you, sir. Am I audible, sir?
Yeah.
Yes, sir. First of all, I would like to thank you, sir, for your for sharing your knowledge with us. We understood a lot about this fruit fly, the distribution and management. Sir, so from our Zoom chat box, we got some of the queries from different persons, participants. That is, first is from Dr. Vijay Kumar, sir. He has asked regarding management of pomegranate fruit fly. Their cracking and whatever the loss is there to the fruits due to this fruit fly.
Okay, in there is a hole that is not with the by the infestation of fruit fly, that is from the fruit fly. Sorry, pomegranate anar fruit fly. That is pomegranate fruit fly. Yeah, that is not fruit fly. That is butterfly actually.
Yes.
Okay, sir.
सर ये फ्रूट
yes.
फ्लाई का ऑर्गेनिक कुछ है नहीं?
ऑर्गेनिक ही बताया है मैंने एक्चुअली. Mass trapping is the organic. You are no need to spray on the fruits.
Yes. हाँ जी.
And if you use in large scale or in your area, there is a large scale cultivation of fruits or that is
Uttarakhand, Bagesar district.
Yeah, so you can use the trap. Trap is not a direct spray on the crops. Just install in the field in the larger number, and you can get the maximum collection. And slowly in 2 or 3 seasons, you can find the drastic reduction in the population.
Okay, sir. Sir, thank you, sir. Sir, one more query is there from Dr. Madhusmita madam. she has asked, is there any ITK method for fruit fly management? And this pheromone trap is the good method, or any other method should be followed like that? She has asked.
ITK method, that is field sanitization. You can go for that, or bagging, because earlier people used to keep fallen fruits. They give to the cattle for feeding purpose. So on that basis, that fruit fly larvae, that is digested by the cattle. So on that basis, we managed earlier. But nowadays, people are not focused on the field sanitization, and collection of fruits are not very good in the orchards. So that's why the population is very high.
Yes, sir. Sir, actually, she has asked this question before you told management, so I hope she understood the things. Sir, next is from Dr. Mahfouz, sir, Mahfouz Ahmad. He has asked, how can we trap orange fruit fly? Any baiting material is there to trap orange fruit fly? Any kind of poison
Orange
bait?
fruit fly, which species actually they talking for? Orange fruit fly is the Bactocera minax. I think if he's asking for the Bactocera minax, there is no attractant for that, or no pheromone trap for that particular species available in the market. But yes, if you go for the mimic of that orange fruit, you just keep plastic orange fruit with the adhesive, or that plastic fruit mimic to the original orange fruit, then you can get the large number of fruit fly adults stick on the sticky trap. In China, there is a huge cultivation of orange and citrus, and they use this technique only. The mimic of they install the plastic orange fruit that is green in color, mimic to the unripe orange fruit, and adhesive on that, and on that basis they manage in the field.
Okay, sir. Thank you, sir. Thank you for your nice deliberation, like for understanding the species difference and the available of management practice. Sir, one more thing from all of the participants and mostly from the students, they are requesting you to provide us with this PPT, sir, your prepared
I've
materials.
already
So
Ritesh, Dr.
yes, sir.
Ritesh, so he may circulate to you all.
Yes, sir. Okay. Thank you, sir. Thank you very much, sir. We learned so much things from you. Thank you, sir.
Thank you so much, Dipan sir. Thank you so much, Prabhakar sir. In such a short notice, you accepted our invitation. I called you in the morning and you agreed. We are very grateful to you, sir, and it was very nice pleasure of mine after hearing you after a couple of years. Thank you so much, sir.
Thank you, Ritesh, and thank you all the faculty members and the Centurion University for giving me the opportunity to deliver a lecture on fruit flies. Thank you so much. Thank you.
There is one announcement to all the participants. The link for our feedback form, as well as for downloading the certificate is available in different platforms like Zoom, YouTube, and in our WhatsApp group. So they can click on that link and give a feedback for this webinar, and also they can download their respective certificates. Now I would like to request Dr. B. Praveen sir, Professor and Head, Department of Plant Pathology, MS Swaminathan School of Agriculture, Centurion University of Technology and Management, for giving the vote of thanks and conclusion of this webinar, this 4 days webinar, which is in the series of faculty development program, which was jointly organized by Department of Entomology and Department of Plant Pathology. Sir, please.
Okay. Thank you, Mr. Ritesh. Am I audible? Hello.
Yes, sir. Yes, sir. Please.
Yes, sir.
Good afternoon, everyone. I deem it a great honor and privilege to propose the vote of thanks on this memorable this occasion. First of all, let me first start by giving my glory to the Almighty God for making this FTP a resounding success. And I would like to thank our management, President Sir, Vice President Sir, Vice Chancellor Madam, Registrar Madam, Deans SP Nanda Sir, Devendra Reddy Sir for their unstinted support for this event. And particularly, I would like to convey my best wishes to the speakers. My heart goes out to thank our respected delegates for accepting our invitation and encouraging the morale of the faculty, students by their august presence and enthusiasm with their insightful talks. And also, I would like to conclude before saying vote of thanks to the faculty and others. Just I would like to give a small gist or summary of the events that had been occurred from 11th. So the first talk by Dr. Sandeep Patra sir, the scientist from Division of Crop Protection, ICR Research Complex. He delivered a talk on organic pest management. Dr. Abhijit Gatak, Assistant Professor, Department of Plant Pathology, Bihar Agriculture University. He delivered a lecture on fungal root-borne disease management using post-mushroom substrate. Dr. Biswajit Patra, Assistant Professor, Department of Agriculture, Entomology, UBKV West Bengal. He delivered the lecture on insecticide resistance, its mechanism and management. Dr. Subrat Datta, Associate Professor, Department of Plant Pathology, BCKV, presented on overview of soil-borne disease with special reference to Sclerotinia sclerotium, Rhizoctonia solani, and Sclerotium ralphsi. Dr. Amalendu Ghosh, Senior Scientist, Entomology, IARI, delivered a lecture on this topical application of double-stranded RNA for management of viruses and vectors. Professor Krishna Kamaka, Professor and Head, Department of Plant Entomology, BCKV, delivered presentation on hidden enemies of rice crop. And today, in the present lecture by Dr. C.S. Prabhakar sir, Assistant Professor, Department of Entomology, Bihar Agriculture University, presented on diversity, spread, and management of fruit flies in India. Thank you, sir, and all the professors and the delegates for their beautiful presentation with their insightful and also the knowledgeable talk, which is very much helpful for all our faculty and the students. My heartfelt thanks to the head of the various departments, faculty for their presence in this event since day one, even with their busy schedule. And particularly, I owe special gratitude to the conveners, Dr. Nihal and Mr. Ritesh Kumar, for their efforts in making this occasion a grand success. And also, I would like to thank all the distinguished faculty of the departments of entomology and plant pathology for their contributions for making this event a resounding success. Last but not the least, thank you, my friends, and all the other colleagues for their cooperation in making this event a resounding success. Once again, I would like to thank all for your patient attention. Thank you. Over to Ritesh.
Thank you so much, sir, for your nice talk. Dear participants, it was very nice and enjoyable 4 days webinar in the series of faculty development program. So we are ending this webinar here. Thank you all.